So moving up to the next session before lunch, we have a session by Dr. N. Bharathan, who would be talking on advances in genomics and gene editing technologies and transforming health. To chair this session, I would like to call upon Dr. C.S. Shastri, Director of Research and Strategic Initiatives, NGSM Institute of Pharmaceutical Sciences, Bengaluru, Karnataka. Sir, kindly come and occupy the chair. Dr. N. Bharathan.
Thank you, ma'am. It's my honor to introduce one of the eminent speaker, Dr. Bharathan. He is an educator with innovative ideas and passionate teaching and scholarly service. His research interests include basic and applied aspects of plant microbes interaction, fungal genetics, and plant and fungal molecular virology. His long-term goals focus on isolation and characterization of virus genes, production of transgenic plants, and identifying extrachromosomal genetic elements from economically important plant viruses and plant pathogenic fungi, and their role in conferring race specificity, pathogenicity, and hypovirulence, and potential use of specific cloned nucleic acid sequences from hypovirulent strains in the biological control of fungus. He is a member of several professional societies, including American Society of Plant Pathology, then Society of Plant Pathology, American Society of Microbiology, Pennsylvania Academy of Science, and Pi Eta Sigma. He has been awarded numerous grants, contracts by various foundations, where he has been the principal investigator for the research. He has a vast number of presentations and publications to his credit. He has also guided around 66 students for presenting various national and international conferences. Of course, he is going to talk on a very exciting area of genomics, and that is, we can say, mother of all sciences, because that is the thing where he can actually go for personalized medicine, possibly drug discovery, and the cure for diseases. Very exciting area we are here, and the organizers will switch on the timer. That's why I will not take much time. Over to you, sir. Please, all yours.
Thank you very much. I'll try my best to make it as simple as possible, and I'll try give you some information with respect to what Dr. Lehman was talking about, with how to go about developing biomarkers and why these biomarkers are so significant no matter what type of biological sciences, pharmaceutical sciences that you're involved in. And let's ask a very fundamental question, because when I did my PhD and postdoc, there was nothing called genomics. But then a situation arose that I was looking at certain types of proteins in a highly pathogenic, plant pathogenic fungus that was totally destroying all the vegetable crops, rice, corn, wheat, potatoes, all over the world. And my project was completely funded by Department of Defense. Why? Because this fungi was infected with a virus, and the virus was lethal to the fungus. So my primary purpose in this project was to sequence the viral genome that is infecting the fungus, and then go about looking at the types of proteins that are formed as a result of interaction between the fungus and the host cell itself and the virus. Then came the entire genomics world, We have sequenced the human genome. We have sequenced the influenza virus. We have even sequenced the pets. And we have gone to the extent that there are several startups all over the world specifically to develop certain types of AI in animals that can detect cancer cells in individuals who are infected with or having an early type of cancer. So some of these AIs are not originating in the United States, in Europe, but they are originating from India. And this is such a powerful tool, and people are already talking about how AI could go and advance genomics, transcriptomics, metabolomics, and any of the omics that you're talking about. And this brings us to a very important component of the pathogen itself. The picture that you see of the COVID-19, this was actually drawn by a computer, and this particular virus, COVID-19, is approximately 38 kilobases long, and it has got some very important markers. And these markers are so powerful that you will be surprised that by understanding these markers and by understanding the mechanism for detection, we have saved billions of lives, not millions, billions of life on this planet. And this has given rise to what we call as the entire genomics component. So what this particular slide is showing you is the plantation plants have been sequenced, the mice has been sequenced, the forest trees have been sequenced, and more specifically, we have very important cultivated crop plants like tomatoes, which have got high levels of alkaloids, have been sequenced and cloned to produce these valuable alkaloids in our body. And then you have the farm animals, the cultivated crops, and pretty much the animal husbandry and the food industry. All of these are now sequenced. Now we have come to ourselves to a very important phase in our lives. And we have to think seriously about public health. You have the mosquitoes, you have the tick bites due to ticks. In the United States, the Lyme disease is a very important disease that is more powerful with respect to the neurological disorders. And in India, you all know that the mosquitoes are responsible pretty much for most of the dengue fever that we have, the Zika fever that we have in the United States, and what we call as the West Nile virus disease that came to the US with the help of the mosquitoes. So the vectors have been sequenced, the ticks have been sequenced, and the pathogen carrying by these vectors are also sequenced. So let us try as a very fundamental unit of what are we trying to achieve. So the common sense prevails that any time a pathogen infects a host cell, the host cell tries to respond to that infection process. It produces different types of proteins. It could be a virus infecting, it could be a parasite infecting, and the ability to detect the pathogen that is infecting and the host, the type of proteins that it is producing, it is very important that we understand the complete sequence of the genetic material, DNA, that is coming from the host cell and of the pathogen, and look at the final product that is protein that is formed. So the proteomics is such a powerful tool moving forward. So let's understand the three basic things that our keynote speaker was talking about: genomics, transcriptomics, proteomics, and the metabolomics. All of these are very important in the pharmaceutical science, in public health, and in the case of the physician. Why is those so important? My previous speaker talked about the biomarker development for Alzheimer's. I will be talking a little bit not about the Alzheimer's, but how to modify the genomic content so that we can find some of the proteins that might be associated with it. This triangle is called as a pestilence triangle. In this particular triangle, you have a pathogen, the environment, and the host. Let me ask this fundamental question to you all. As a pharmacist, as a drug maker, what are you trying to do? You are trying to prevent the pathogen infecting the host cell. So by doing that, you're modifying the environment so that the pathogen cannot infect the host cell. What does the pathogen do? The pathogen tries to undergo mutation. It tries to undergo modification. It tries to undergo or produce certain types of proteins that can literally prevent the host cell from reacting to it. So this is a very dynamic process, and in this dynamic process, humans play a very important role, the pathogen play a very important role, and then you have the host cell playing a very important role. With this, we started a project. It is a plant pathogenic fungus, folks. Pretty much the total loss of the crops worldwide is approximately $1.5 billion. That is a huge sum for a country like United States, India, and the European countries to be looking into the pathogen infecting rice, wheat, and corn. So what we did was we used this fungal virus model system. You can see here from my postdocs and PhD work, that is a virus particle approximately 30 nanometers in diameter. And all the viruses infecting this fungi are double-stranded RNA genome. And the double-stranded RNA is very unique that you all understand that influenza virus and, for example, the viruses related to COVID, we had several strains of the COVID virus. We had several strains of the influenza virus. So we are asking ourselves this question. How come you have different strains of these RNA viruses? And likewise, in a virus that is infecting the pathogen, like the fungus, we have different viruses infecting the Arsolani fungus. So our hypothesis is serotype variation is common, so likewise the biotype variations must also exist. So with that in our mind, we took up an international project of sequencing the entire pathogenic fungus. Believe it or not, folks, we sequenced this in 2014. The total cost of this sequence was only about $500,000, and you can see Barathon is still the fourth author providing all the money for sequencing purposes. Why? Sequencing is a club. Only certain group of individuals can sequence a very important humans, pathogens, or no matter what. So the individuals, Dr. Neerman belongs to JCVI, John Craig Ventry Institute. It is the same institute that sequenced the human genome. So you have a situation wherein a Barathon working with a plant pathogenic fungus, you have a human genome project, Dr. Neerman, involved in sequencing this fungus. This fungus is approximately half the size of human genome. It has got 12 chromosomes, and it has got close to about 20,000 genes. So now, it is very important for us to see what type of proteins are formed by this fungus. Now, for all of you students who are here in pharmacy and medical school, I just want you all to know from your basic biochemistry that isozymes are very common, right? You all have studied a lot about isozymes. You all have studied about isosugars, glucose, fructose, sucrose, and all of these disaccharides that exist in aldehyde form, keto form. You have even isogenes that are involved. So for the first time, we saw isogenes in a eukaryotic system. So you have a pathogen producing, expressing certain types of genes at certain times during the infection process, but never expressed during the later part of the day or later part of the infection process. This tells you the complexity
of the TCA cycle. The TCA cycle is a very common cycle, folks. After glycolysis, you have TCA, and after TCA, you go for what you call as the ATP production phase, which is very important. This is the same TCA cycle. Look at the complexity involved when we go for sequencing and finding some of the very important glycosylated proteins that are involved, carbohydrates that are involved in this infection process. So we have gone and cloned the gene, we have sequenced the gene, and we got all of these products. So now we went to the next stage, looking at the proteomics, that is what happens during the infection process itself. Dr. Lehman talked about the processes involved in the proteomics itself. I have somebody's phone ringing. I apologize. So we looked into, after the genomics component, we looked into the 2D gels, and we looked at the peptides that are involved, and we use a lot of the mass spec and mass spec analysis, machine learning, and bioinformatics. This is the general workflow, and I assured all the pharmaceutical science students that if you want to do anything in genomics or proteomics, that should be the workflow of the project itself. We have put the nucleic acid on a chip, we have put the protein on a chip. All this is based on the chip analysis of nucleic acids and proteins, such a powerful tool. We need 1 nanoliter of the purified protein. Then we did some 2D gel analysis and followed by a mass spec. And you can see here, folks, the complete sequence of the protein itself. All the colors that you see here are nothing but the amino acid molecules that are involved. And you can see some of the variations in those single amino acid sequence. All these are publishable results. And we ask this fundamental question, serotype variation is common, suggesting biotype variation could also occur. So that's about the RNA, that's about the protein. So we did what you call as a decider analysis. When is a protein expressed? When is a protein not expressed? You can see here, this is an infected tissue, this is a healthy tissue, low level of expression of the protein. This is 100% healthy, this is a totally infected tissue. You can see a higher level of gene regulation and the protein that is formed. So we do a lot of analysis of the cider, looking at the expression and non-expression of these proteins inside a cell. That brought us to a very important next stage, that is developing biomarkers using the very important assay techniques. And I just want you to know at this point that this is how my project was funded by, all over, from McGill University to the NC State Raleigh, North Carolina, to Department of Defense. Then I want to spend some time talking about genome editing. Why is genome editing become such a powerful tool? Genome editing involves nothing but engineered nucleases. It is so powerful a tool, folks. You will forget for the next 5 to 10 years that we did certain types of biology, and that biology has become so irrelevant today. All of you must have learned in your high school biology and in your college biology that these are nothing but knockout mutation, insertion, and we call point mutations. All of these are powerful tools, and these types of tools exist in nature, and man has modified the changes in the DNA molecule using these types of mutations. But what is important now today is that it is so precise that you can actually tailor some of the genome modification itself. This is the title of science that carried. You can modify the B you can modify any organism that you see today, folks. We have total of 1,200 biological organisms on this planet that has been genetically modified, edited by CRISPR technology. And two individuals, Jennifer Donda and Miss Dr. Carpenter, she's now working with Max Planck. Both of them got the Nobel Prize for their work in 1917. Now, interestingly, you will find Fan Zhong. He's from the Broad Institute at MIT. He's being referred to for the Nobel Prize, but he did not get the Nobel Prize. That is his team. And if you ask the question, where is the boss in this? And you can see the boss is just sitting there, and the rest are all PhDs and postdocs and technical assistants working in this lab. So the entire nature carried what is the role of the PhDs and postdocs in CRISPR. It is so powerful a tool, folks. You have a woolly mammoth that was discovered in Siberia. You can actually recreate, bring back to life a woolly mammoth using this type of gene editing technology. Never take a bacteria lightly. Bacteria gave us endonucleases. Bacteria gave us CRISPR. Bacteria is unicellular, folks. They are existing for the last 5 billion years. But this clustered, regularly interspaced palindrome repeats. CRISPR, as it's called, is a type of immune system function in this unicellular organism. So this unicellular organism protects itself against any type of bacteriophage infection. You all will be surprised that why did Jennifer Donda and Dr. Carpenter get the Nobel Prize? You all must have heard in your high school biology, in your college biology, that proteins, DNA polymerase, RNA polymerase, all these are regulatory elements. For the first time, they showed that the RNA is a regulatory element. So we now know that RNA plays a significant role in cutting this DNA molecule. Like any other gene editing protocol, the Cas9 system is a protein. That is the RNA, and the RNA has got signals. This is called the PAM signal, and it cuts the DNA molecule exactly 3 nucleotides ahead of this PAM sequence. So you can do gene disruption, gene modification, and gene correction. All of these can be done, and one very important mutation that has been created because of this is I will come back to this in a couple of minutes, sickle cell disease. There are people now in the United States walking with complete cure from sickle cell disease. I just want you all to know, folks, many of the technologies that we apply today is tested on mice, is tested on other organisms. Today, CRISPR is tested on living individuals. They are not dead. So you are actually protecting the life of the individual as they are living. So we talked about drug discovery. Drug discovery takes approximately, you start off with 100,000 or 1 million compounds, and by the time you come down to one compound, it takes 10 years, 15 years, 20 or 25, or sometimes you do not get anywhere close to it. So what is important is that CRISPR allows for drug discovery to speed up the process as quickly as possible. This has greatly reduced the cost itself. And the next important thing that I want you all to know is, in CRISPR technology is so powerful that we developed in our lab a mechanism by which you could detect the virus. So what we did was, we hooked up the virus to CRISPR modified polymerases, and we had all the 3 genes that we could include with that CRISPR. And what we did was we used the same types of probes that is essentially used, and this is allowing us to detect up to 2 virus particles in the body of an individual. That's how powerful this tool is. So for us, virus detection is another very important tool. This is not my field, folks, but I want you all to know that in the futuristically, we had Dr. Lehman come to us from St. Louis, Washington. So that lab there has developed several T cells called the CAR T cells. And the CAR T cells are so powerful that you can actually take the normal T cells from the individual, genetically modified by CRISPR, and develop a cargo. This cargo has got unique proteins that can detect several types of cancer cells, and you can reintroduce those back into the body of the individual. This is a very powerful immunotherapy, and it has been so powerful that developed not in my lab, but a different lab at UPenn. They tested on 4 patients, and all the 4 patients were absolutely phenomenal. They stayed only for 2 weeks, but the data that they got from that immunotherapy was very powerful. So the take-home message here is there are several ways by which you can modify the cell using CRISPR system. The most important thing I want you all to think about is the chimeric antigen receptor T cell. That therapy is most advanced, and pretty much there are many labs in the US doing this, and all you have to do is take the healthy T cell, develop a cargo in which you can introduce the modified T cell and then re-inject it back into the humans, and this pretty much is going to be released into the market. Believe me, folks, in another year or 2, Amazon will be selling CAR T cells. All your primary care physician has to do is he'll make a phone call. Oh, Barathon has got B-cell lymphoma. Can you send CAR T cells for this? It is delivered within 24 hours. You go to the hospital, take a shot, walk out. That is how powerful this immunotherapy is. Most importantly, India is going to be right in the cusp of things. Why? I showed you the gentleman from Broad Institute. All he has done is he has got CRISPR modified for all the 1,000 genetically understood cancer cells, and he's looking for ways by which these cells could be multiplied. And Dr. Lehman talked a lot about the tau cells. These are all specifically NTLA amyloid cells. They use lipid nanoparticles. Very powerful tool, folks. If you want, you can, in India, the amyloids is a genetically transmitted disorder. So you could get CRISPR-treated TTR-specific guide RNA. That's how it is introduced. That is pretty much a rectified protein that does not form the amyloid structures. There are several labs developing this protocol using knockout, insertion, and consecutive editing. So your basic genetics becomes very important for you all. And one last thing that I have to tell you, if liver cells regenerate and you have a CRISPR-edited liver cells, can you in perpetuity maintain the CRISPR edited T cell is a question this group is asking, so you can actually have CRISPR regenerated T cells. So I'm going to stop at this point. Why? Because the transfection techniques are basic techniques that all your lab has to learn. You all cannot give up basic biology, folks. Basic cell biology, go back, study hard, because these types of mechanism of DNA repair in our body is endogenous. You do not do anything. All you do is a repaired, CRISPR-edited DNA molecule that you introduce, and that becomes a major tool for gene modification. Any questions? Whew, I'm tired. Guys, you gotta go ask questions. This is a class that takes me 2 semesters to teach. And I have more than 100 students from undergraduate, masters to PhD, and many students from Arya College in Mumbai and St. John's School of Pharmacy who are taking these classes. Why? Conventional pharmacy is gone. It will cease to exist if you are looking at molecular biology based pharmaceutical science. You have to go back to basic biology, understand this, and come forward, because that's how futuristic pharmaceutical science is. I've ruffled some feathers here, but I cannot. But that is the truth. You have to accept it, because you all have to understand higher education is not easy, it is expensive, and you have to value what you're spending. Yes?
Thank you for the wonderful presentation, sir. I want to know what is the status of this gene editing. Are there any treatment regimen for sickle cell anemia or thalassemia minor?
Yeah. So the question is, folks, is gene editing toxic to human bodies? Pretty much. I've not shared all the slides here. What happens when you're doing TTR studies, most of the engineered cells are put into mice, okay? But the take home here is mice is not shown any level of toxicity. And I also told you that only 4 individuals were tested on this particular study. And all those 4 individuals had terminal cancer stage 4. So when FDA approved the use of the gene-edited T cells, the reason why that type of permit was given was to study toxicity. Was not to simply study the cure. There is no cure at stage 4, yes. But the idea is that if the same edited T cell was given to individuals at stage 1, surely they could have shown progress.
So the toxicity was not there?
Right. Absolutely zero toxicity.
Thank you, sir.
Yeah, thank you. Thank you, Dr. Brarthan. I think you completed your talk.
Yes.
Yeah.
For me, it was more students
four
asking
more
me
minutes are
questions.
there. I think it can be utilized for the question and answer session.
Yes.
Thank you very much for that wonderful, a bit scary lecture, in the sense that I have a question, sir.
Yes.
See, with this age of artificial intelligence or general intelligence and your genomics and other things, well, it is good for drug discovery or disease discovery as well as personalized medicine. All those things are fine. But what about the ethical side of it and the regulation side of it? If the things go into a wrong hand, see, you can actually generate superhuman beings, super things, imbalancing the nature. That's what the concern what people will have if it goes to a wrong hand. If the research goes without any bridles, then what will happen? in future what may happen. We have seen it happening in Manhattan research.
Yeah. So the question is, what if gene editing goes to wrong hands and it could be lethal to human beings? Well, this can happen any time, right? So any type of tool created could be lethal to anybody. So I don't think science has to control that component part of it. So it is very hard. What science is telling you is, yes, the tool is available. How do you want to use the tool? Are there ways by which you can actually cure an individual and improve the quality of life of an individual? I say this with utmost confidence, folks. The reason for that is you all have learned C4 photosynthesis and C3 photosynthesis in your basic high school plant biology, right? You have corn, wheat, rice, barley, and all of these are what type of plants? Let's go back and answer this question. These are all C4 plants. All the lawn grasses that you grow here are C4 plants. So what has mankind done over the period of thousands of years? We have selected only C4 plants. What do C4 plants do? They are able to convert and release the atmospheric carbon dioxide. But C3 plants cannot do that stuff. They utilize the carbon dioxide from the atmosphere. So there is a difference. So what we have done is, by selecting a C4 plant, man has indirectly added to the carbon dioxide content in the atmosphere. So we are playing a big role. There comes science that you can actually convert some of the mechanism of cultivation process so that we use sustainable crops so that we do not have this competition of introducing carbon dioxide into the atmosphere. Folks, Elon Musk is thinking big. It is absolutely phenomenal. Going back to your question about AI, I'm going to take this laser thing and point it to this little blue line that you see there. That little blue line is such a powerful tool, folks. It is what is responsible for insertion. It is what is responsible for gene knockout. It is what is responsible for deletion. So, if you are so precise in those gene modifications, and it is this PAM group of 3 nucleotides, don't tell me God made this. No. These 3 nucleotides are A, C, G. Adenine, guanine, cytosine. That's it. This is such a powerful tool. You can make this cargo in the lab. It is just a 1-hour reaction at 37 degrees and reintroduce this modified T cells. So the perception that AI will kill us, the perception that this genetic tool will kill us, it is all hard to sell, very difficult to sell, because mankind is not ready to really understand if we can live with an AI which is a superhuman to humans. Can a superhuman dictate to humans? We do not know that. That is what we have to fear.
Thank you, Dr. Bharatan. You can see a great scientist as well as a passionate teacher in him, and a wonderful lecture. Thank you very much, sir.
Thank you.
Any questions? No, I think we are running out of time. If you have got any questions personally, you can interact with Dr. Bharatan. Thank you very much, ladies and gentlemen.
Thank you, sir. Be here, sir. Thank you, Dr. Bharatan, and thank you, Dr. Shastri, for chairing this session. I hope this was an informative talk, and some of you have gained certain insights into the talk. So, to thank our speaker, Dr. N. Bharatan, I'll again call upon Dr. Pushpa Sarkar to kindly give a token of appreciation to Dr. Bharatan. Thank you, sir. I would request ma'am to kindly give a token of appreciation to our chair, Dr. C.S. Shastri also. Thank you so much, everyone. Now we are going to break for lunch. As you can see, we are running late by 1 hour. So we'll not have 1 hour lunch break. Let's cut it short by 15 minutes. We will have a 45 minutes of lunch break. And we'll start our session try to start at 2:40. So please be here. All the delegates have your lunch and please come back before 2:40. We are going to start the session then. Thank you very much.